And yeah, thanks everyone for joining our monthly webinar series, which is part of the UK Freshwater Quality Research Programme, which has been funded by NERC and DEFRA. And here at Leeds, we are supporting the programme. And obviously, we're really looking at freshwater quality, particularly at the sources of pollutants, the cocktail of pollutants that gets into our rivers, and the impact of these pollutants on our freshwater ecosystems, and how, in addition to all that, we're experiencing climate change and land use change. And so we're looking at these things as well. And really the aim is to improve the ecological status of UK rivers and inform better adaptation and mitigation of these risks, and obviously have an impact for policy as well. And within this programme, there are 5 projects. And this year, we've been hearing from each of them. And this year, it is the turn of quantum. So I'll pass over to Penny later. But, there are 5 projects and you can see what they're all about. And they occur across the UK. And then if you want to contact us, the champion team, there are a number of different ways. You can email us at freshwater@leeds.ac.uk. We have a website, and at this website you can also see all the recordings of our previous webinars, and you can follow us at Freshwater Qual 1. So as I said, this is a monthly programme. We will break in August for the holidays, but next month we're hearing from the 5th project in the programme, which is LTLSFE, that is hosted at UKCH. And then we're going to have a talk in July from Professor Alison Dunn at the University of Leeds on invasive non-native species and freshwater ecosystems. And then, as I said, we'll have a break and we'll return in September with another talk from Laura Carter at the University of Leeds, looking at exposure in the aquatic environment following antibiotic manufacturing. So I'll hand over to Penny now, who's going to tell us more about quantum.
I think I'm off mute now, aren't I? Is that showing as a full slide, Pepper?
Yeah, that's great. Thank you.
Thank you. So thank you very much for the opportunity to present an overview of the quantum project today. In quantum, what we're doing is we're quantifying the nutrient enrichment, pathogenic and ecotoxicological impacts of livestock farming on UK rivers. And we have a big team with a range of very complementary expertise that's going to deliver this programme. And of course, our range of project partners, which are at the bottom of the slide, and then a whole range of organisations who've expressed an interest in keeping track of what we're doing and engaging with us as we move through the research programme. So those are our stakeholders. And if you're from a stakeholder organisation that is not on that list at the moment, then please do get in touch with me. My email is at the bottom there, and we will ensure we engage with you in our future communications. So livestock farming is a key driver of changing quality in UK rivers. And if we look at the graphs on the right, you can see that most of the livestock in the UK are in the north and west, where we have wetter, heavier soils, we have more rainfall, we have steeper slopes. So we tend to find that's where we find most of the cattle and the sheep. And our programme is focusing specifically on cattle and sheep, not on pigs and poultry, because it simply wouldn't be feasible within the budgets available to have incorporated those as well. That's not to say they're not also really important contributors to the changing quality of UK rivers. So cattle and sheep farming is concentrated in these areas. It's on about 10 million hectares of grassland, which represents over half of all UK agricultural land. And that might come as a surprise. You often see programmes about farming on the television, and you see it seems to be all about the arable, but actually a very large amount of the land is grassland. And on this land, there were in 2020 nearly 10 million cattle, nearly 33 million sheep, and collectively they were excreting 4 times as much nitrogen and 3 times as much phosphorus as people across the whole of the UK. And again, with the focus on sewage and sewage discharges, you might be forgiven for thinking that that's the major cause of declining quality in UK rivers. It's certainly a very important cause, but it's not the only one. The livestock are also important. 80% of the estrogen in the UK environment derives from livestock, and cattle excrete an order of magnitude more estrogen than humans, particularly dairy cattle. Livestock-derived pathogens, veterinary pharmaceuticals, and other ecotoxins, they're processing the critical concentrations at which they have impacts on the stream biota actually remain to be established. We actually don't know this information yet, nor do we really understand how these interact then with the livestock-derived organic nutrient loading. And the role of the environment in modulating the way in which these impacts occur is also unknown. So the Quantum programme has been set up to address this knowledge gap, and it's focused around 3 themes. And these are the main themes of the overall freshwater quality programme that you've been hearing about in this webinar series to date, but fitted to our livestock focus. So in theme 1, we're exploring how the climate and catchment changes affect the sources in work package 1 and the processes in work package 2, by which these pollutants from livestock farming enter water bodies, are mixed, are transported, and then leave the river ecosystem. Under theme 2, we're then investigating the impacts of the mixtures of these chemical and biological contaminants from livestock farming and how they generate exposure regimes for the riverine biota. And we're looking in particular in work package 3 on the ecotoxicological impacts of this material, and in work package 4 on the nutrient enrichment impacts of this livestock excreta on the stream environment. And then finally, in theme 3, we're using our work package 5 to develop pan-UK advice on livestock as a driver of changing quality in UK rivers. So it sits alongside the very strong knowledge we have about other factors such as, for example, sewage systems. So just to give a little flavour of what we've been doing, work package 1, we've been defining the character of the different livestock excreta, and our research questions are to determine what the chemical and pathogen composition of this material is. It's our source term, it's the stuff that's going into the water bodies, and then how does its composition vary according to the way in which we produce or manage livestock within the landscape. And so our approach is to collect representative samples from sheep, dairy and beef cattle farms. You can see my lovely team here engaged in doing that, and they're all smiling. So they seem to be enjoying the work despite the nature of the material. We're focusing on on our university and research platform farms at the moment, and also with one local farm that's very close to our field platform to give us local information. And that does tend to mean that they're then working with very healthy animals. And that does have an impact on the initial results we're getting in terms of looking at pathogens, for example. We're analysing the samples when they come back to the lab to determine nutrient profile, organic matter composition, the ecotoxins, so these are the chemicals that cause toxic effects on the biota, a range of compounds called fecal biomarkers, which are only found in ruminant digestion, for example, and therefore when we see them in the river, we know they've come from livestock and not from sewage. And then the pathogens. And again, some of those pathogens are specific to livestock farming rather than to human sources. Samples are also being sent to an organisation called NRM Kwood, and that's going to then allow us to compare our results directly with results for lots of farms across the UK under what's known as RB209, which is the Nutrient Management Guide applies across the UK. And then from this, we're then going to be identifying a suite of organic nutrients, ecotoxin and pathogen targets, so focusing in on a specific set of chemistries that are representative of the range of chemistries that are there, which we're then tracking through all of our experiments that are running in work packages 2 to 4. So this is where we are at the moment. We've completed the autumn-winter 2023 sample collection, and we'll be going out again in the summer and July, August of this year to collect a second set to try to capture variability in the farming cycle. And these are the farms that we're visiting. So there's Henvice Farm, which is in North Wales, and it's run by Bangor University. That's a sheep farm from which we're able to get urine, dung, and farm up and use samples, because of course slurry isn't part of the sheep farming production system. Ibers Aberystwyth, Harper Adams University, Rothamsted, North Wick, these are all big research platforms. Windhurst Farm, which is the University of Bristol's veterinary school farm, which is very close. It's about 1.5 miles from our major field site where we're doing our work package 2 experiments, which you can see here in yellow. And then Eberlands Farm, which is a very similar farm to the farm where we're doing our experiments. It's a low intensity beef cattle grazing, external grazing farm. So we've got 40 samples in total. And it's just going to give you a flavour of the types of data. I can't possibly show you all of it because we're collecting a lot of information. But this, for example, is some of the information that we've got from NRM Kwood. So you can see, for example, that the pH of the different types of manure from dairy, beef and sheep is very similar. But if we look at the dry solids content of the manures, and the urine and the dung and so on, then we can see that we have much higher dry solid content in the sheep than we do in the beef, and that's higher than we do in the dairy. Now, the sheep, of course, that's partly influenced by the fact that there isn't a slurry sample in the mix there. If we look then down at the total nitrogen, the total phosphorus, particularly with the phosphorus, we can see that the sheep excreta contain more phosphorus as a weight percent than the beef, and the beef and the dairy are very similar. And with the nitrogen, we're seeing a similar sort of trend there. So it gives you a flavour of the range of data that we're now starting to collect. And then we've been doing our own experiments to explore some of the more detailed chemistries. And this is just some early data for the inorganic nitrogen concentrations. So this is nitrate, nitrite and ammonia added together, phosphate concentrations, and then we're also able to measure urea directly. And as you can see, for example, if we focus on the phosphate concentrations, we have the lowest phosphate concentrations in our urine and the highest ones in dung. And if we look at the urea data, you can see that we have the highest concentrations of urea in urine, unsurprisingly, and then we have the lowest concentrations of urea in the dung. So the excreta are all different from each other. So what we're doing now is we're doing pathogen screening for a range of viruses and bacteria. We're doing ecotoxin analysis with screening for over 300 compounds, and then we're looking at the dissolved organic matter chemistry and pre-screening for tens of thousands of compounds in these samples, and then using that information to identify our targets. And I'm just going to let you have an idea of the types of analysis that we're actually doing now. So our work on the pathogens is being led by our team at the University of Bangor. So Dan Davis, Davy Jones, David Chadwick. And this is the approach that's been adopted. So we have sample collection, which you've just seen. The samples are then concentrated and the material is extracted. And then the RNA, DNA content of those samples is quantified using something called qubit before it goes through a qPCR run to identify the viruses or bacteria that are present. So this is the list of the ones that we're zooming in on. We have E. coli, which many people will have heard of in the context of sewage, but it's also a really important reservoir of this within cattle in particular. It's also found in sheep excreta. We've added in a human biomarker just to check that we aren't getting human signals through those materials as well. So if we see those, we know we've got some kind of, perhaps some septic tank effluent mixed in with some of those samples. Then we've got a range of adenoviruses. We've got the tuberculosis bacterium, chlamydia abortus, which causes spontaneous abortion in mammals, and then viral diarrhea. So this is an example of the type of data that we're getting. We've got lots of data. We're just starting to work our way through it. So this shows the concentrations of DNA and RNA in the extractions. And you can see, for example, here we've got quite variable results. So we've got relatively low results for the Harper Adams dairy farmyard manure, but much higher results for the dairy dung. And there's the beef farmyard manure, the sheep farmyard manure, and then the sheep dung. And this is the result of a qPCR detection. And as I said before, we're focusing on these farm platforms where we tend to know precisely the disease status of the animals at a fairly high frequency, and we're only allowed to sample healthy animals. So at the moment, we've only picked up the E. coli, but we have seen that then in all of the farms that we've actually visited. So the E. coli is present all year round. In terms of the ecotoxins, this work is being led by the University of Bath, Tolulope Lawrence, Tolu, and then Barbara Kasprzyk-Horden. So the ecotoxins that we're monitoring are shown in the full list. And then those that we've actually detected in samples from just the slow samples from just 3 farms. So Aberystwyth, Windhurst, and Harper Adams, that's where we've got to at the moment with those analyses. Those are shown in blue. So here on the antibiotics list, you can see that around about a third to a half of the compounds we've screened for have been detected in that material. Endocrine disruptors and hormones, we're seeing those are appearing probably about 60 to 70% of the compounds we're screening for are appearing in those samples. Fungicides, antiparasitics, so far we've only found 2, but we don't have methods for the ones in green. So we're actually developing methods for those at the moment. And then we have the pharmaceuticals and biomarkers. And again, it's around about 50, 60% of the list that we've looked at has now appeared in our samples. So we've got around about 60 samples, 60 targets in total, have been identified out of screening for 344, and there's more methods being developed at the moment. But perhaps what's more interesting is we are not the only project under the Freshwater Programme that is looking at the ecotoxins, and Ecomix and MOT for Rivers are also looking at them. And the ones that are ringed in red here are the ones that they're focusing on, often in relation to sewage sources, but also in relation to other livestock and animal sources, so dogs, flea treatments on dogs and so on. And you can see that there are some where they're interested in the compound, like trimethoprim, but we're also interested in it because it's appearing in the livestock excreta. So there's opportunities to explore synergies as we go through the programme later on. Finally, moving to the methods for the livestock-derived DOM, which we call LDOM, which we're characterising in all of the excreta, river water and biotic samples in work packages 1 to 2 and work packages 4 to 5. This work is being led by the Organic Geochemistry unit at the University of Bristol, Victoria Hussey, Ian Bull and Richard Evershed. So the way we're approaching this is we're using a complementary suite of methods to reveal this chemodiversity of this organic matter pool. And I'm showing you here data for one site we've sampled in a previous programme I led called Merck Domain. A couple of papers there which are free to download from my personal webpage if you want to read them later on. And here what we're using is a range of complementary methods to expose the different compound groups. So in the first one, we can look at polar compounds. We have untargeted analysis with direct infusion mass spectrometry. What does that mean? It means we haven't got the names of any of the peaks there, we've just got the peaks. We can see that there's a lot of information in there. If we try to estimate how many peaks there were, it was quite difficult to do. So we can zoom in and you can see that even within this blue window, we've got a lot of detail there. And if we zoom in on the purple bit, there's even more detail available. And in fact, in that one sample from a dairy farming catchment, there were over 19,000 organic matter compounds in the stream that are derived from that livestock farming practice. We can also look at other compound groups, so non-polar compounds. So for example, here we're able to look at some of the lipids, and without going into the detail, you'll be able to look at it in more detail later when the webinar is finished. A lot of the compounds there are directly related, for example, to grass and animal feeds. So here we can see our urea, for example, in that sample. And then we can look at macromolecular compounds, so things like lignin and lignin-derived products using pyrolysis, GCMS. So that's what we're doing with the samples. It takes a long time to do, so there'll be a time lag before we're able to actually present those data for you. So moving on to how we're actually doing the experiments, this is work package 2, where we're investigating how manure management, environment and in-stream processing transform the DOM pool. So we're looking at how the material transforms once it arrives in the stream through in-stream processing. How does it change in relation to variation in hydrology and climate? How does it change in relation to how we manage things in fields? And then what happens in terms of the exposure rates for the stream biota under the different flow and management? So we have our experimental catchment here, which is in North Somerset. We've got a very nice pair of farmers here who've rented us out a large chunk of their field. We are doing an extreme experiment between sites 1 and 5, and then we're doing an experiment in the field itself between sites 5 and 8. And the critical thing with that one is that most of the catchment upstream from the experimental areas is non-agricultural. So with the stream experiment, the idea is that we sample upstream from a dosing bar. We then dose the chemistries into the stream that we see in the livestock excreta as identified in work package 1. And then we sample the stream as we move progressively downstream over a maybe 300 to 400 metre reach, and we see how that chemistry has changed. And then in the field experiments, starting strangely with strip 2, strip 2 represents current practice under current UK government guidance. So we are asking farmers to keep their livestock 10 metres away from the stream. Typically that's done with fencing, which is funded under government schemes. So we're going to be looking at the effects of that on the stream between sites 7 and 8. But then we wanted to say, well, would it make any difference if we moved those strips further away from the stream? So what if the strip was 40 metres away from the stream? So in each one of these, we're applying slurries, and then we're measuring the effects of that on the stream. So just giving you an idea about the sensitivity of some of these methods. Before we started the experiments in the village and down in the field, we collected some samples at site 1, which is the top of the stream. Site 2, outside the pub, it's around about 20 metres further downstream. Site 5 is about 250 metres downstream and forms the entry point for the field. And then we have Site 8, which is at the bottom of our experimental field strip. And the field is low intensity beef cattle grazing. So it's external grazing, but there is dirty water spread by a sprinkler in the yard. And you can see that while the chemistries are moderately stable as we move through the village, with the total concentration not exceeding about 520 nanograms per litre, we have a big step change to around about 800 nanograms per litre when we get into the field. So that's adding another 300 nanograms on top of background, which would be 0 for these compounds. And one of the causes of that is this bisphenol A, which is a contaminant that's largely unlikely to be derived from the breakdown of agricultural plastics. So it's just to give you a flavour of the types of information that we're generating. In terms of the stream dosing experiment, we've found having a lot of fun with this over the last month or so. We're trying to work out the right way to do the dosing, and we still haven't found the right way to do it, but we will work it out. And once we have our technique sorted out, we're potentially going to take the technique out to around 6 sites around the UK, probably in Wales and England, because we need to be able to get the samples back fresh to the laboratory before they start to decompose. And if anyone on the call has got suggestions of non-agricultural catchments with small streams where we might be able to do this, please do get in touch with us and give us your suggestions. Moving on to the field experiment. So the field we've got is a gentle slope from the top on the right here to the bottom, and you can just see the stream on the left of the photograph, top left-hand corner there. It's a fast-flowing stream. It's got a lot of plant life in it. And what we've been doing is trying to establish how this system works, because there is nothing worse than setting up an experiment and putting your sampling equipment in the stream, only to find that there's a hidden culvert, another subsurface structure that's diverting the flow, not from the fill slope down to the stream, but further downstream where it's not being picked up by your samplers. So a huge amount of work has gone into that. We're working closely with the Lancaster University team, led by Andy Binley and with John Ball, and then my team with Anna Castro-Castellon and Sydney Enns. So I'm not going to go through the detail on this slide. I'll just tell you a little bit about what that has shown us. So the topographic survey, so looking at the slope of the land, has given the black lines on this chart. The rainbow colour, which stands out really strongly, is telling us about the conductivity of the soil. And based on this information, we have established 2 plots. So here's our 40-metre plot, which is on average 40 metres from the stream, because streams are never, well, not rarely straight. And this one is 10 metres from the stream. And we have 3 flow samples at the toe, where we're collecting what's coming down the slope before it arrives at the stream. And then we have our sampler at site 5, where we've got an automatic water sampler. We've got sondes measuring a range of chemistries every 15 minutes, and also a water level recorder. So we're able to establish the flows through the stream. Site 6, compared to site 5, allows us to pick up the residual background influence of that grazing regime with dirty water before we added our slurry. Comparing site 6 to 7 allows us to then look at the influence of having that slurry amendment on the field, 40 metres from the stream, and then comparing sites 7 and 8 allows us to compare whether or not we're getting more flux when the livestock are able to get closer to the stream. So we are right at the start of this experiment. We started the amendments back in April. We're putting 7 amendments on in total, ending in October, in compliance with guidance. And in total, we'll be putting on a total of around about 250 kilograms of N per hectare, as is the advised rate. This is the type of system we've got. It's precision application. It's taken a bit of trial and error to work it out, but we've got it right. Here's my lovely team. They're still smiling, despite the fact they're going out there selecting and collecting the slurry. And our monitoring for the first experiment actually started just yesterday. And we'll be doing that for 4 weeks and then repeating monitoring in October. Moving on to work package 3. We have the team is led by Charles Tyler with Dania Albini and Hannah Boot at the University of Exeter, and they are literally starting the experiments as we speak. So their questions that they're answering are: what are the toxicities of the key compounds that have been identified by BARF in the excreta to the freshwater biota? What are the bioaccumulation rates and the effects on invertebrates and fish? And how does the ecological toxicological impact on the biota vary according to environmental character and function? So I'll just take you through an outline of the experiments. So they're going to be using Daphnia pulex in single colonies and then Gammarus pulex. This is the Daphnia is the water flea, Gammarus pulex is freshwater shrimp. It's very abundant in the site that we're looking at and around the UK. And we're not measuring chemical responses to this dosing. We're measuring the biological response. So things like the number of newborns, the number of the brood, the presence of males, body size, and the rate of death in relation to the stresses that are presented singly and then progressively in combination. And these are the target compounds we're looking at the moment, which you might recognise from the earlier list. Moving on from that experiment and learning from the outcomes, we're then going to test the effects of 2 of the target chemicals singularly in combination at the community level. And the experiments will include multiple trophic levels, so from phytoplankton to zooplankton to macrophytes. And we are still not sure whether those will be done under controlled conditions in the laboratory or whether they'll be done in mesocosms in the field. So things we're going to be testing will be things like the bioaccumulation of the chemicals in the organisms, survivorship and abundance, body size, and then looking at molecular markers that indicate chemically induced stress in the organisms. And then finally, and I think this is going to be great fun, in the autumn, they're going to be coming out to the field with us, to our field site, where we're going to be doing the Gammarus again, but also adding the 3-spine stickleback to experimental treatments in cages in the stream. This is going to be chronic 30-day dosing. And again, we're looking at the same sorts of things in the Gammarus, but we're looking at exotoxins accumulating in the liver, in the stickleback, and also liver transcriptomes. Finally, moving on to the last set of experiments, we're moving our focus in work package 4 from ecotoxins to the role of this material as a nutrient resource bioavailable to the freshwater biota, which we previously established in our NERC domain programme. And the questions we're asking are, how does uptake vary between different livestock excretory and chemistries? How does its bioavailability vary according to environmental character and function? And how is uptake modified by the matrix in which it appears, so an acid stream versus, say, a chalk stream, and the interacting stresses that are present in the livestock excreta. So we're using a technique we pioneered in the domain programme, and you can see here us applying it in a small peatland stream in North Wales. We have a series of mesocosms, each is about 6 litres in size. We fill them with the river water. We add pebbles that are coated in the epilithon or biofilms you may be more familiar with. We collect bryophytes, mosses or liverworts, and we wire them onto chrome nuts and we drop them into the bottom of these jars. And because these organisms don't have roots, they're taking nutrients up through the water, so they're able to respond to that dosing. Then we collect the samples over a series of time points and then send them off to our colleagues at OGU for analysis, where we're going to be looking at both the bulk and compound-specific nutrient uptake, and using that to trace trophic transfer from primary producers to consumers. So we use the bulk, the dual-labelled isotopes in particular, because it allows us to check to see whether the molecule is being cleaved, which is usually what happens with urea, and then either the nitrogen or the carbon is taken up, or whether the entire molecule is taken up intact. Target compounds for this experiment will be based on those found in the work package 1 samples. And just to give you an idea of where we are with this, because we're planning to start this experiment in June, we're going to be using urea, which is commonly found, you saw in milligrams per litre levels in livestock excreta, a couple of amino sugars. Again, we've used the glucosamine before, which comes from fungal cell walls, but we're also interested in moranic acid, which is associated with bacterial cell walls. We're quite interested in using creatinine, which is excreted by kidneys as a breakdown product from protein metabolism. We've used glutamate and glycine before. Glutamate in particular has been a particular focus for our early work. And then we're going to do an amino acid mix. So we're not just exposing the organisms to one compound, but to a range of different tasty compounds that they can utilise. And then in order to compare the rate of uptake with what you might get from inorganic nutrient forms, we're also providing 15N nitrate and 15N ammonium sulfate. And just as a spoiler, I can tell you from the previous experiment, we found that these compounds that we've used previously were taken up at least at the same rate as the inorganic nutrients. These are bioavailable, highly dynamic chemistries that we see coming out of these types of farming. So last bit then, work package 5 is where we pull this all together, because you may have been sitting there thinking, goodness, there's a lot of different experiments going on. How's that going to turn into something useful? So what we're going to be doing is exploring whether there are generic livestock impacts on UK rivers that are linked to specific livestock types or environments, how those impacts might alter in response to a changing hydrological cycle, and whether we can produce transferable evidence that underpins policy and management. So we've got a couple of slides on this. We have a second tier of sampling, which is what we call tier 2, where we will be going out and collecting and analysing water samples in the same way you've previously heard, for 20 sheep farming sites, 20 dairy cattle sites, and 20 beef cattle sites in comparable environment types, to find out whether the information is generic. So if we're always seeing urea, we're always seeing glutamate at particular concentrations in a particular site, or ivermectin, do we always see that every time we have sheep farming, for example? And then we are then going to build a model for our work package 2 site using all of the information that we've collected from the work packages 2 to 4, using that information to parameterize the model. We can validate that model against our tier 2 sites using the tier 2 published data and also partner data, including from other freshwater programme projects, as that's available. And then we'll be developing scenarios and testing those in consultation with our partners and our stakeholders. So do let us know if you'd like to be involved in that. Also in alignment where we can with the scenarios that are being run through other projects where modelling is taking place. So just a little highlight then of how we're doing the tier 2 sampling. We have a national biogeochemical modelling framework, which I developed with my colleagues under the NERC Environmental Virtual Observatory Programme, which basically says that if you do something in a particular way in a yellow area, you will see the same response if you do the same thing in other yellow areas, where yellow in this case is our upland catchments. And it's a very powerful tool that's worked extremely well in previous work. And there's some publications where you can find out about that at the bottom of the slide. So we're going to be going around the UK. I will tell you, these squares are just plonked on the map at the moment. We haven't decided exactly where we're going yet. So again, if anyone in the audience has got ideas of sites we might go to, obviously we'll be going to other freshwater programme sites as part of that collection. Please do let us know. So just to sum up then, what do we expect Quantum to deliver? These are the highlights. We're going to be characterising the nutrient, pathogen and ecotoxin contaminant mixtures exported to UK rivers from different types of livestock excreta in work package 1. Work package 2, we're going to be showing how that material behaves in the environment and what that means in terms of exposure regimes for the stream biota. In work package 3, we're going to be exploring the ecotoxicological impacts of this material as this varies according to environmental character and function. In work package 4, we're looking at the nutrient enrichment effects. And then in work package 5, we're exploring how our work can deliver through modelling and upscaling this pan-UK understanding of how livestock farming drives the changing quality in UK rivers, both now and in the future. And to find out more, we have a website. You can follow us on Twitter, or you can email me directly and I'll pass your query on to the most relevant member of the team. Thank you.